We are going to go over topic 1, structure and function. A lot of this has to do with the human body, but you will see that you can also apply this to other organisms, in particular plants. The first thing that we always start with is the standard. The reason why we're starting with the standard is because this is going to be how the questions are framed on the Regents exam. Develop and use a model to illustrate the organization of interacting systems that provide specific functions within multicellular organisms. Right below here, I have all the human body systems. You should be familiar with them and their general function. They might get more specific with what they do. Hopefully, whatever is more specific will be in the reading, but we're going to try to just give a general overview for each of the systems. First up, we have the cardiovascular system. I always like to think what's the major organ in each of the systems, and looking here at the cardiovascular system, you will see that we have the heart as well as blood vessels. So right there I have my heart. Instead of the cardiovascular system, you also could call it the circulatory system. That brings us to a word that we need to know, circulate. When something is circulating, that means it's moving. And the things that are moving inside of your body, you have the heart that acts as a pump, and it pumps the blood around the entire body. Now, we're not so interested in the blood, but what is inside of the blood? What's inside of the blood? Well, that's oxygen, carbon dioxide, glucose, and hormones. So for this, we have circulates. And circulate just means moves. Oxygen, CO2, glucose around body. I mean, this is how water moves around your body. Hormones, which we'll talk a little bit about on the next page. Next up, I've got my digestive system. Digestive system starts in the mouth. It's one long tube. It goes down into your stomach, then to your intestines, and eventually out the body. Again, just one tube from the mouth to the rectum. The function of the digestive system is to break down food into smaller parts. When you eat something, right, that's large macromolecules. Macromolecules include proteins, starch, more complex sugars, maybe like lactose, sucrose. It also could include lipids. Lipids are just another name for fats. We need to break those down into the building blocks. Building blocks of starch are simple sugars. Building blocks of proteins are going to be amino acids. So the word digest really just means breaks down food into smaller products. Now, the reason that it needs to be broken down to smaller products is so that diffusion can take place. Now, we don't need to get too specific into what diffusion is, but we want to remember that diffusion is talking about the movement of molecules without needing any sort of energy. The movement of molecules without needing energy. So let's just highlight those two words. Circulates, meaning move. And then diffusion, talking about moving molecules from high to low concentration without using any energy. You go and you break down food. So let's say you have proteins. Those get broken down into amino acids. Amino acids are small molecules. They're able to pass through the intestines into the bloodstream. That passing through is called diffusion. Next up, we have the endocrine system. Endocrine, you'll notice that there's a few glands that are located here. We have our adrenal glands, which are responsible for making adrenaline. I'm pretty sure that these glands here are supposed to be the sex glands, which would be the ovaries and the testes. The pancreas is another endocrine gland. You have the thyroid, which is located more in your neck. You have the thymus. All of these are going to be examples of glands. I don't think you need to know the specific names of them. They most likely will be in the reading passage, but you should be familiar with what the function of the endocrine system is. So the endocrine system is going to secrete. Now keep in mind, that's another one of those words that we should know, but sometimes it's helpful if we write down a more common word to help us figure out what's going on. So it's going to secrete or release hormones. On the next page, we're going to do the example of insulin, but I'm just going to write that down here as well. So hormones are chemical messengers that cells release to communicate or talk with other cells. The pancreas releases insulin to talk to muscle cells and to talk to fat cells and tell you those cells to take in glucose. The specific mechanism of that we're going to go over in a little bit. Our next one is the muscular system. The muscular system is just in charge of movement, and also it generates heat. Next up, we have the nervous system. Again, just going back, because I didn't do this for the last few, but we always want to think, what is our main organ? The main organ for the nervous system is going to be the brain. The nervous system is in charge of maintaining homeostasis. Sometimes they're going to write this as responds to a stimuli. When it's responding to a stimuli, again, that's really referring to the fact that your body is trying to make sure that it's staying balanced, and that is what homeostasis is. Reproductive system, we have a whole separate section in these notes, but the big idea for the human reproductive system or reproduction system is that it's going to produce sperm and egg to make an offspring. Then we have our respiratory system. You'll know that the respiratory system has the lungs. The lungs are responsible for gas exchange, so it takes in the oxygen and then it's going to release the carbon dioxide. Skeletal system is going to be our support. And then finally, we have the urinary. The urinary has been on quite a few exams so far. I don't know if we'll see it again. We just want to remember that's the removal of metabolic waste. And it's also going to be in charge of osmoregulation. Osmo is referring to water, and regulation refers to control. That's our human aspect, but what we have to remember is on the test, they can give you any example as long as they provide you with the information in the passage. So yes, we might get a human body system. If you get the human body systems, make sure you know how two of them interact. They might kind of force your hand into what two systems you pick. Therefore, you should know all of the systems. Two good ones to always pick would be the nervous system, because that has the brain and that controls all the things. Also, if you pick the circulatory system or cardiovascular, that is going to be a good choice because the blood circulates all the stuff. It picks up the oxygen, circulates that. It picks up the hormones, it circulates that. It has the water. It brings the water to the urinary system where it gets filtered out. It takes the food from the digestive system and moves it around. All right, on this page, now we actually have the plant. I took this right from the Regents on the left-hand side. The right-hand side is from an old Regents. It gives me my systems. Just like humans have different systems, plants have different systems. You have the root system and then the shoot system. The top part that's above the soil, is the shoot. And then the part that is below the soil, that's the root. From what I've seen on the test so far is if they're going to give you something like this, they're going to provide you with all of the evidence that you need, so you don't really have to go looking. It might ask you how two systems interact. You've got to make sure that you take from the root side and you take from the shoot side and you write in a sentence how they are interacting. Keep in mind the root system. What's going up? Water. What else is going up? Nitrogen. Remember, nitrogen is the nutrients in the soil, not food. I know that gets confusing, but the nutrients from the soil are really like vitamins and minerals. I don't know why I wrote the S. So we've got water goes in, nitrogen goes in, and then if I'm thinking about my shoot system, I got the sun, sunlight, the leaves take in the sunlight. The other thing that we have is carbon dioxide enters from the underside of the leaf. One thing I do not have in this packet that we're going to want to add is that the shoot system contains guard cells. Oh, I have that down below. / stomata. That's actually how the carbon dioxide and the oxygen get in and out. There are tiny pores on the underside of a leaf. And while plants don't breathe how we breathe, right, they have these tiny little pores, and the pores are large enough so that oxygen and carbon dioxide can move in and out. And again, that's on the underside of the leaf. The top part of the leaf has a lot of chloroplast that allows the plant to trap the sunlight or capture the sunlight so that photosynthesis can take place. And notice I drew all these arrows. Sunlight goes in, that's an input. Carbon dioxide goes in, that's an input. Water goes in, that's an input. Now we're switching back to the human side. Disease is a failure of homeostasis. I haven't defined homeostasis yet anywhere, but when I'm thinking about homeo and then stasis, that prefix homeo or the prefix homo always means same. And then stasis, we're talking about staying. So we're trying to stay the same no matter what's happening. We've all been sick. First thing it says, immune response. When you are sick, your body produces white blood cells, and those white blood cells make a protein called an antibody. So here's what happens. You're sick. A germ comes in. You could call a germ a pathogen. You could call it a microbe. You could call it a germ. I mean, something that makes you sick. So here I have my germ. On all the cells, my cells, your cells, plant cells, animal cells, doesn't matter, all the cells, viruses aren't cells, bacteria, all of the cells have antigens on top of their cell membrane. And what those antigens are, those are ID tags. And when your white blood cells are in your blood, they're cruising along inside your blood vessel, and then they see something that doesn't belong there. How do they know it doesn't belong? Well, those cells have these antigens here. Once the white blood cell recognizes, oh my goodness, there's an antigen here, and that antigen doesn't belong, the white blood cell, which can look kind of like a little Pac-Man, the white blood cell starts making, right, here's my white blood cell, Pac-Man. The white blood cell starts making these antibodies. Think back to protein synthesis. proteins have a specific shape. Notice how my antibody and my antigen seem to have matching shapes, like a lock and key, a specific shape. Sometimes it'll be called a complementary shape. Now this antibody can fit right onto that antigen. Right? They have that same complementary shape. That's going to allow the antibody to potentially break open that germ, and then it cannot do any more damage to your body. Now, once you make those antibodies, you've got memory cells. Your body will remember that you have been sick with it. So hopefully you don't get sick again. Or if you do, maybe not as bad. Now, some diseases you do get sick with more than once, and you might be like, well, why is that? You see those antigens? They can change just a little bit, and then your body is like, oh my gosh, I've never seen you before. It's kind of like an old-timey show where there's someone who's running away from the cops and puts on a little fake mustache, and then the cops don't know who they are anymore. Same idea. The antigen changes just a little bit, and your body is totally confused. It's like, I've never seen that guy before. And you're like, oh my God, it's so similar. Of course you've seen it, but you haven't. So then you have to make totally new antibodies. And that's why you get sick again. How have we tried to make it so we don't get sick anymore? Well, different technologies. One is going to be a vaccine. A vaccine is a dead or weakened pathogen, or it has part of a pathogen, and it causes you to make antibodies, right? Because you inject a little tiny piece of the virus or the bacteria, and then your body has an immune response. But the stuff that they put inside of you doesn't actually make you sick, or maybe makes you feel sick for a couple of hours, but you don't get the full illness. The other thing which has really decreased disease or the length of that someone's sick with diseases would be antibiotics. Antibiotics can only be used to treat bacterial infections, but they help in order to make you feel better quicker. The next standard is all about feedback mechanisms and homeostasis. This goes back to that lab that you did, the state lab, where you put your hand either in something that was hot or something that was cold, and you recorded your extremity temperature, meaning your hand temperature, but you also recorded your body temperature or core temperature. And what you want to see is if your hand was in something cold, did your core temperature change? If your hand was in something hot, did your body temperature change? Right? What happened to that core temperature? First up, we've got feedback mechanisms. It says maintain a living system's internal condition within certain limits and mediate behaviors allowing it to remain alive and functional even as external conditions change within some range. Again, we have that word homeostasis. The more you practice splitting apart the words, the more intuitive it becomes on the test. So if you've never practiced that before, now might be the time so that when you get to Regents Day, you are just killing it and you know exactly how to split the words apart so that they make more sense. We have two types of feedback, a positive feedback and a negative feedback. Positive feedback encourages what is going on inside of a living system. Think about you turned in an English essay and your teacher kept writing about how your sentence structure and your topic sentence and your evidence was amazing. That means the next time that you submit an essay, they expect to see the same thing. So positive feedback is encouraging whatever is going on to keep on going, right? Keep that up. You're doing a fabulous job. That's what positive feedback is. We have 3 real-life examples. One is root development in response to water levels. What winds up happening is roots grow underground, as we saw in this picture. And as they start growing down and they get closer and closer to the water source, it's like, oh my gosh, we're getting more water. And then more root development happens closer to the water. And then more root development happens closer to the water, right? That's a positive feedback because you keep growing closer and closer and closer and closer to the water. So therefore, you can get enough water to survive. Blood clotting. Blood clotting is going to be when your body releases platelets. Platelets are responsible for clotting the blood. So you release platelets, then platelets wind up releasing chemicals, and when you release chemicals, you attract more platelets, and more platelets means more chemicals, and then eventually you'll get a clot wherever you're bleeding. Someone in one of my classes said, well, blood clotting isn't good. That's what causes heart attacks to develop. Think about that when you have a cut and you're gushing blood. Blood clotting in that case is good, right? So think about it more like a cut. You have a cut or you have a bruise inside of you. Platelets go to the scene. They release chemicals. More platelets come. It makes like a little internal, not external. We're talking about like an internal scab. Childbirth, same thing, positive feedback. When you're in labor, you release hormones or secrete, right? Secrete could be another word. When your hormone level increases, you have contractions, which is when the muscle tenses up and then relaxes, tenses up, and then as you get more contractions, your hormone level increases. As your hormone level increases, you get more contractions, and so on and so on and so on. So positive feedback is like, yes, you are doing an amazing job. Keep it up. Keep going, keep going, keep going. Don't stop. Negative feedback is different. Negative feedback is like, you are doing a terrible job. You must stop right now. So positive encourages, negative discourages. Please stop what you're doing. We need to get back to our normal value. Some of these we did in class. One, heart rate response to exercise to adequately transport oxygen and carbon dioxide around the body. We did some exercise. We took our heart rate. We saw if it increased or decreased. What we saw was that it increased. That's in order to ensure we're getting enough oxygen to our cells because those cells at the time need to make more ATP. Other one, stomate response. Stomates are the space. If you're looking at a leaf, right? Here I have a leaf. On the underside of the leaf, there are little tiny spaces that we can't see there. A lot of times I'll draw it like this. This being my top of the leaf, this being the bottom. In this little space, which is the stomate, allows for carbon dioxide to go in and oxygen to go out. So guard cells, and they're not guarding from bad things. They're guarding so that you don't like take in or let out too much oxygen, carbon dioxide, and water. So guard cells surround the stomate or stomata. On a really, really hot day, if it's really hot, you want to conserve or save water. And if this is one of the first videos that you're watching that I've done, make sure, these fancier words, if you don't know them, make some flashcards. Knowing those words are going to allow you to unlock some of those passages that the Regents are going to have, right? Some of those cluster questions that have the confusing wording. The more you practice getting down the regular vocabulary, the easier the questions will be. On a sunny day, let's say it's not too hot, but it's sunny. It's like 70, perfect, sunny out. They might actually be open to let in more CO2, because if you have more CO2, then you have more photosynthesis happening. Next example is body temperature. We've all been hot or we've all been cold. So we've got sweating and then we have shivering. And the last one is blood glucose levels. That's going to be on our next page. This was just on the January Regents of 2026. So I'm thinking you're not going to see it, but just in case. We have a feedback mechanism and what it looks like when we see a graph. Notice this is a negative feedback. Remember, negative is like, stop what you're doing. Things are not going well. If we don't get back to normal, we're going to wind up dying. First thing we have is this line. That line is the set point. Your body wants to be at the set point. That's the happy line. Let's say over here, this is blood sugar. It could also be called blood glucose. First thing you'll notice is that there's a spike. So think to yourself, why would my blood sugar go up? That should be pretty straightforward. That could be because you're eating. So right here, I have my stimulus. And my stimulus in this case is eating. Your blood sugar goes up. That's normal. Everyone after they eat, their blood sugar goes up. But then your body's like, we're supposed to be kind of like in a really tight range. Things are getting a little crazy. We're getting a little bit too high. So what does your body do? It creates a response. So the stimulus is the factor that's making the change. And then up here, I have my response. And in this case, this is when pancreas releases or secretes insulin. And insulin's going to lower it. So I've got my increase after I eat. Then I make insulin. Insulin brings my blood sugar back to that normal number. Overnight, you're not eating. Well, ideally, you're not eating. It's like, maybe you eat dinner, you have a snack at 9 o'clock, which is kind of late, but maybe that's when you're eating. And then you don't eat again until the next morning. Some of you might even skip breakfast. When you're not eating, this stimulus, not eating, is called fasting. Now, you don't want to be releasing that insulin because your blood sugar is going to go even lower. There's a different response for this one. The same organ makes it. Now the pancreas makes glucagon. And glucagon is going to cause that number to go up, right? Your blood sugar to go up. And again, we're trying to stay in this tight band. If somebody has type 1 diabetes, they don't make insulin. And since they don't make insulin, their blood sugar stays elevated. People with type 2 make insulin, but their insulin does not work. Looking over here, this is from the Regents exam. They might ask you how multiple systems are interacting. I look here, I see I have a blood vessel. Again, what I would say is you want to label all the things if possible. So a blood vessel is your circulatory system. Because if it says system, you can't write down blood vessel. You have to write down the name of the system. I know picky, but make sure you've got that. Here you see stomach. You might want to be like, stomach, I'm good to go. Nope. Make sure you write down what the name of the system is. Over here you have pancreas. Can't write down pancreas. Have to write down endocrine. I mean, pancreas is also part of the digestive system, but in this aspect, we're going to say endocrine. Notice here, they do label it. They tell me this is insulin here. It's also insulin here. Insulin is a hormone. The hormone gets released or secreted into the blood vessel. And then eventually, it's going to make its way to its target cell. In this case, the target cell is the muscular cell. Remember, we'd have to write down muscular system. And once you have that target cell, when the insulin and the receptor match up, that's going to unlock this portal, right? That glucose transport protein, and the glucose that was inside of your blood from your digestive tract now can enter into the muscle cell. Once it's inside of the muscle cell, the test might want you to make all these connections. That's going to be when you can have ATP being made. CO2, sorry. So we have oxygen plus glucose gives me ATP plus CO2 plus H2O. Down here, I have vocabulary that's in the curriculum that you should know. Some of these words you'll see repeat themselves later on, other ones maybe not. First up, we have a prediction. A prediction used to be called a hypothesis, right? It's what you think will happen based on your research or based on evidence that you currently have, and then you're going to make a prediction, and maybe your experiment will not support that. Independent variable is the thing that I myself am changing. What is the difference between the two groups? So I am always thinking independent. You might also see something written as the effect of blank on blank. The first blank is always my independent variable. The second blank is the data that I collect, or the dependent variable. A constant's anything that's staying the same in the experiment. Constraints are going to pop up in a lot of other areas as well. The constraint is a limitation. Usually we're thinking when we do an experiment, the cost, the safety, the materials, how much time we have to complete it. Criteria are requirements. Sometimes you'll see constraints and criteria overlap. We've got precision is a measure of how close a series of measurements are to one another. Precise measurements are highly reproducible, even if they are not the correct value. So you keep not getting the bullseye, but you keep shooting the same spot, not the bullseye. Then that is going to be really precise. Accurate is you're on the bullseye. The last two are quantitative and qualitative. Quant means quantity. So quantity is referring always to a number. And then right below that, we have qualitative, qual, like quality. So an observation that you could make. That is topic 1. So you're going to want to make sure that you review the vocabulary, you know about the human body systems. If they give you a different type of system, I think most of that is going to be provided to you, like what the functions are. You might have to make some connections, things that we did not specifically go over. On the first exam, they talked about red blood cells. Students had to know that red blood cells carry oxygen. It was in the passage that they carried oxygen, but it was not in the section that you needed. As I have said, or will say if you're watching the videos in a different order, you need to ensure that when you're answering a cluster, you might have to go back 2 pages to find what you're looking for. It could even be on the next page. So make sure you're using the test. You might have to flip around to find it within that cluster, but if it says something about evidence, it's there. It just, sometimes you're on a scavenger hunt in order to find all of the evidence. As always, if you have any questions, feel free to write them and I will try to answer them to the best of my ability.